Hello, everyone. Thank you for joining our webinar today. I'm excited to have Joelle here with me hosting the webinar. My name is Amy Knock, and I will be moderating today's session. Before we begin, I just wanted to let you know more about how this webinar will run. The webinar is being recorded, and the slides and recording will be available for you within 3 business days. A short Q&A session will follow the presentation to answer any viewer-submitted questions. During the webinar, you can submit your questions you have using the webinar sidebar menu. Select the Questions tab, type in your question, then hit the Enter key on your keyboard. Remember, you can submit questions anytime throughout the webinar. And that concludes my very brief introduction today. Joelle, I will now pass it over to you.
Thank you, Amy, and thank you everybody for joining. Sorry, losing my voice already. That's not a good sign. But we're going to talk today about microbial stress, growth, and produce testing, and in generalities, how bacteria adapt in our food samples in our system. So with that, we'll get going. So the next thing, apologies if you can hear my dog in the background. That is not cool. Company overview, just if you're not familiar with Eurofins, is that we're a global leader in biological testing with over 200,000 reliable analytical methods. We're all over the country. I think we have over 800 laboratories globally in 50 different countries and over 50,000 employees. We do a lot of different testing. That's within the food space as well as environmental and other testing industries. So if there's literally something to be tested, we probably test for it. We do over 400 million different tests annually. So it's a little bit about Eurofins if you're not familiar. We also have certification and auditing arms of our businesses as well if you're looking for that. Today, what we're going to be talking about is microbial stress responses, viable but not culturable. So you may have heard this acronym, the VBNC, in your past industry experiences. The impacts of testing of those stress responses that it may have on your actual testing programs in the food industry, and then a little bit of an explanation about microbiomes and microbial dynamics in respect to these stress responses and how it can be a useful tool for us. So we'll kind of start at the very beginning if we think about origins and just bacteria in general. So they've been on Earth for at least 3.5 billion years, so pretty much they're the life forms that we expect or think we have on the planet. And they also continue to dominate when it comes to what species are actually on the planet. So we've got 78% of bacteria, of all the species that are known, 78% of them are bacteria. And then you look at kind of the breakdown here, so animals are actually only 7.3%, and the like plants, 1.02%. So effectively, just a lot of abundance when it comes to bacteria, and that leads to a lot of different types of adaptation to live all over the planet, right? So they survive in all locations on the planet, including places like Antarctica, where it's really, really cold, not a lot of accessible, free water, hot springs and geysers. We get a lot of actually of our PCR genomic tools for some of the enzymes of these organisms. You can find stuff in Old Faithful, like that. Anaerobic location. So we often think about oxygen being something that's really necessary for growth, but certainly not the case with microorganisms, and they can grow perfectly fine, many of them, in anaerobic locations. So ultimately, kind of just the take-home point is that they're highly responsible to all different types of environmental, highly responsive to all the different types of environmental conditions. They are rapidly monitoring these conditions and can have very fast, rapid changes to make sure that they survive. So they become experts at survival. We'll go into a little bit more about that. And they have quite a multitude of different responses and mechanisms to protect themselves in different situations. Just as an example, you may have seen in the news in the last year, really, is that TITANIC is being eaten by microorganisms, and this is I guess significant because it was previously thought that the depth of this shipwreck would preserve it. So between the pressure of how deep it was, how cold the environment is, how little oxygen, etc., that it would just be preserved in this state for years to come for us to analyze, right? It sunk 110 years ago for those of you who aren't Titanic experts. So it's been there quite a while. But now what they're actually finding is that there's these metal-eating bacteria that are consuming 100 kilograms of iron per day, which is quite a lot. And it's estimated that by 2030, so not too far from now, is that they will be fully consumed by these microorganisms. So just a case kind of study about how adaptable and how different, right, these microorganisms can be. We would never imagine being able to survive off of eating bacteria, certainly not at those depths. Surviving off of eating the iron like these bacteria are, and certainly not at those depths. As you can just see really the evolution of how many different bacteria species there are and what they can actually evolve to basically be able to optimize for survival. So when we think about these bacterial origins, really what we can think about is this phrase, right? What doesn't kill you makes you stronger. Bacteria have figured out how to be very creative and adaptive in all the environments that they live in. Those could be temperature related, UV, very, very low water activity like deserts. You'd also have very high UV activity as well in this situation. Ice, so generally that's how we think to preserve things, but there's definitely organisms that can remain viable and growing in very, very cold conditions. Or even salt flats, so very, very low water activity due to a high amount of salt, but they can develop mechanisms to be able to survive in those situations. So effectively, all of these challenging environments become selecting for survivors, right? So they have developed these mechanisms to be able to survive and really thrive in all these different conditions. Many of these responses are pathways to repair, damaged DNA. So if they get exposed to high UV and they have errors in their DNA, they've developed systems that will rapidly repair it, that upregulate different genes that they may have and that allow for different defense modifications. One example of those things would be heat shock proteins. So they're actually proteins that are intended to help with other damaged proteins. So potentially if you had a change in temperature that caused you to lose the form of a protein, which is its function. If you remember, kind of from your biology classes, it's form equals function. Is that if it gets heated and you start to lose some of that form of a really critical protein, there's actually other proteins that have different structures that will help get them back into the right format so that the cell can still maintain its processes. You can also have more significant modifications to proteins and lipids. So if you've been evolving in a different environment, say at high salt or cold temperatures, that are more long-term modifications. So if you look at thermophiles, the organisms that like heat, you'll actually find that their amino acid breakdown in their proteins have increased ionic bonds and hydrophobic components that actually help the protein maintain the structure that it needs for its function, even at those higher temperatures, whereas if an organism is not set up to be living at those higher temperatures, their proteins themselves will denature and they won't be functional. Another example with halophiles, so salt-loving organisms, is that you could have what they call compatible solutes that actually are produced by the organism to increase the osmotic pressure inside the cell so that it matches the external, so you don't have any issues with osmosis actually destroying the cells. You can maintain your functions. Psychrophilic, so cold-loving, they have more pockets of space to minimize ice formation in their cell membranes as well as proteins, and it helps them maintain fluidity, even though things around them might be very stagnant because of the cold temperatures and frozen situation. Another term that we've been hearing a lot more lately about is viable but not culturable, or viable but non-culturable, so VBNCs. We talk a lot about it, but what really is this and what does it mean to us in the food industry? Effectively, it's a survival advantage to harsh conditions. So that could be nutrient starvation, extreme temperatures, changes in salt or pH or osmotic stress. So those things change over time. Oxygen availability, perhaps a lot of it or not enough of it. Intense light exposure, pressure, heat, chlorine, that's definitely something you see a lot in the food industry. And effectively what it does is enters a dormant state or a hibernation state where there's very limited metabolic activity for these organisms. That limited metabolic activity, so they're not like rapidly growing or not doubling at all potentially, is also makes them somewhat resistant to some antibiotics because they're not bringing things in and out of the cell, or different types of antimicrobials. One of the key things about the VBNC state is that it often cannot be cultured using traditional culturing techniques. So say growing them up, perhaps we have a standard protocol, we follow it, and then within 24 hours, let's say the 24-hour incubation, there's nothing there, but perhaps they're still there, they're just not rapidly growing. There's evidence that cells can be resuscitated to full metabolic activity. So in recent research, researchers have found that there are ways to actually activate these cells and bring them back into a rapidly growing state where you would start to recover them. At this point, it's something that's being studied a lot and certainly talked a lot about is, what is the actual impact on food production system, this VBNC state, and what does it mean for our testing? All right, so what's the relevance to the food industry? Well, I guess unfortunately or fortunately, depends how you want to look at it, the target organisms that we look for in the food industry have been shown to have very robust stress in VBNC states. So these are Listeria species, monocytogenes, Salmonellas, E. coli, pathogenic E. coli, so these would be like your O157s, your S-tex, your Shiga toxin-producing E. coli, Vibrio species, Campylobacter, Pseudomonas, so common spoilage organisms, and Shigella. So unfortunately for us, and obviously this is not a comprehensive list, but these are the ones that are often most relevant when it comes to food safety, is that as we've been learning more about stress responses and organisms and their adaptations, is that many of the pathogens that we're looking for have very robust abilities to survive, which is what you'd expect, right? That's what bacteria do. They live to survive and replicate, and they do it pretty well. So the main point, they're very adaptable. We've evolved to survive, and that our food systems can present very challenging environments for microbes that actually can induce some of these states and create survivors, right? So what doesn't kill them makes them stronger. In some cases, we can be creating those systems that are consistent, over time, where we actually could be selecting for these survivors. So just very briefly and very top line is that stressed and injured cells may have slower growth rates, which is kind of common sense, right? If they're not rapidly growing, or if they're stressed out or injured, they're going to be a little bit hopping along as opposed to running. So the lag time that we see in the growth kind of curve can become longer. So perhaps, maybe it's normally 30 minutes or an hour with E. coli. Maybe if we've really injured the cell, it's going to take the cell longer to recover to that stress, and maybe it'll double that time. It can also change the optimal growth rate. So once we start rapidly growing, maybe it's not as fast of a rapid growth as it could have potentially been before, because of the conditions that the organisms are in. I'll show you some examples of that in a little bit. The microbiological methods that we use in the feed industry are designed for target organisms, right? That's pretty apparent. If we're doing a salmonella test, we're looking for salmonella. And what does that mean? Well, we generally, at this point, have medias that are growing up these cells and selecting for these organisms that have selective agents that actually help impede other organisms, give them a better shot. Maybe we have temperatures that either make it so that other organisms can't grow, or that we are optimizing conditions for a certain organism. We can add inhibitors and antibiotics, again, for selecting. And we certainly can change biological conditions with different oxygen or different stirring or shaking, or those sorts of things to help for growing. We also can design methods to help resuscitate injured or dormant cells. So potentially there's a less pressuring environment, so they could do a non-selective media and then go to something that's selective. Lower temperatures, we could start them out with something that's a little less intense, maybe room temperature, and then go up to a higher temperature to actually select for it, just to help recover cells that may be not in their optimal state in the environment that we're first finding them in. It's always important to remember too is that you know, the environments that we produce food in are quite diverse, and you're going to expect to see cells at different stages. Some cells may be rapidly growing, and some cells may be more injured. And you can think about it, you know, if you've just done in your environmental, your sanitation program for your cleanup, you've got a lot of chemicals out there that they may be less able to come out of that quickly as opposed to if you took a sample during production when there's not anything out there that's stressing it as much. So here's some examples of just growth rate changes. So on each of these graphs, the y-axis is the maximum growth rate, so how fast it doubles effectively, and just presented in different ways. So these are three different conditions that so salt concentration or water activity is in the upper left corner, is the first one, where we can see that as salt concentration increases, the maximum growth rate actually goes down. So nothing else is changing. This is with Listeria, same medias and same temperature, but as there's more salt, the organism can still grow, but it is obviously a significant decline in how fast it can actually double. Similarly so with pH, if we look at the next top right graph, this is maximum growth rate. It very much is dependent on pH. So at the very, very low pHs, we'd see almost no growth, which is what we'd expect. And then you start to see a very rapid incline of what that maximum growth rate is at the more neutral pHs. And then a more gradual decline as we get into the more basic pHs, where it's still able to grow, but obviously not as well as it can at the 6 to 8 pH. Again, that was the E. coli. The final graph that we have here is the maximum growth rate with different sugars or different additives. So pyruvate being a molecule in the biochemical processes, or acetate. And you can see that that can actually change the growth rate as well in a media. So there's a lot of different approaches when it comes to taking samples and also the media optimization to grow them that can be really looked at to be able to select conditions that can impact growth rate to get to the basically the detection time that we're looking for different organisms. Another example would be Salmonella growth variability, and this is from a paper from Food Microbiology, and it studied 60 isolates of Salmonella enterica in different serovars at varying pH and water activities, and they did that with salt concentration. Basically to just be able to understand better the maximum growth rate and how it can change over time. And so we looked at this, or they looked at this in regards to the changes between serovars, and with Salmonella, there's over 2,500 different serovars of Salmonella enterica, and to see how it changed amongst them, and also then within the serovars. So that maybe it's the same type of murine, it's all type of murine, but different strains of it, to see how much it changed within. Then they basically monitored that growth rate observation and then incorporated that for modeling and method development. And what we saw, I mean, really the take-home point from this study in this paper was that you saw a pretty big difference between pHs and salt concentrations of how much variation there is to that growth maximum growth rate. So somewhere between 6 to 24%. So really just the important thing to think about is that with our methods, we often talk about things or organisms as if they're one group, but there's actually a huge amount of variabilities within the group, just like there is within people. Is that, we could say on average people are, 5'7", 5'5", but obviously it's a bell curve and there's a lot of differences of how tall someone might be. And this is very similar to what we would see with Salmonella or bacterial growth, is that you may actually see a lot of variation amongst strains or even between one strain. So we already kind of touched upon this, but the relevance to the food industry and what does it mean? So common processes that may induce these states or stress responses for bacteria that we're looking for. So pasteurization, obviously that's a pretty intense practice. Some organisms completely eliminated. Some may be actually getting that constant stress that makes them adapt to it. So things to think about, probably more from a spoilage perspective. Chlorine washes and dips certainly are going to stress out organisms. Fast temperature shifts. So if you go quickly from low or high temperature, Bacteria have to respond to that. So if we think about like those heat shock proteins or modifications to their cell membranes and basically overall function, they have to react, and that reaction is not necessarily instant. Increased salts and preservatives, those are going to make it just harder for them to live. Increased pressure, so if you think about high pressure processing and some of the new approaches that we might have to different pasteurization or kill steps. Unique produce industry considerations. Obviously, I'm a produce person, so I think a lot about what could stress mean in the produce environment, as opposed to if we think about like a dairy or a powder facility. We lack control over a lot of different variables. So there's weather events, we have exposure, desiccation. So we give some examples of that. We have some pictures. You think about how dynamic an environment might be for produce and how little control we really have over it, and what the organisms to survive in these environments might do. Certainly, if we have a more controlled processing plant, we probably could model, or at least try to model, a lot of the variables and what the conditions might be for microorganisms at different stages in the process. But in the produce environment, so much of our environment is outside, and we have very little control. So you could have flood events or temperature changes, certainly bird activity or animal intrusions. We do chlorine wash flumes all the time, wind pressures, relative humidity changes, and those could be very microclimates. Those could also be macro, where you have it in huge areas. Agricultural water, cold temperatures, anaerobic, depending on how much it moves, and sediments and those sorts of things. Unfortunately, people might notice some pictures of the fire. That was a fire in Salinas that we saw not too long ago, in the last couple of years. That leads to just a whole bunch of new pressures, different volatiles in the air, obviously smoke. What can that do to different bacterial responses? UV also changes, and ozone changes, and obviously desiccation. So, when we think about happy environments, water is generally a pretty critical component of life. And depending on where you're growing, depending on the crop cycle or the irrigation cycle, you could be putting these organisms through different stresses, where you have a lot of water and you have not a lot of nutrients. So the produce environment by itself can create an enormous amount of different stress responses that are honestly being studied quite often right now by many researchers to see how bacteria might behave in these environments. So all in all, right, it varies day to day within process, by supplier, by their location, by weather, etc. And what's really important when we think about our food safety plans, and we'll kind of transition now into kind of thinking about testing with it, is to consider your product and your process. We talk about that in terms of verification of methods and really designing a food safety plan. But we really also need to think about how your specific process may impact testing or trade-offs that may happen. So if you think about that, is there a difference between speed and sensitivity? Everybody wants a faster method, but the faster you go, you lose sensitivity or the ability to detect, right? And I think if you combine what we talked about in the beginning, is that sensitivity is going to be somewhat related to how fast we can grow something up. And that something is going to be related to how stressed that organism may be in the environment where we find it. So we can talk a little about that. We can budget for stress and resuscitation. So how well does our method help an organism recover? And really, to start to think about, we talk a lot about like matrix verification for testing, but also think about your process and whether or not you're creating a system or maybe experiencing systems that may make your product or sample a little bit more unique compared to maybe other people's. So are you selecting for conditions that would allow an organism to adapt, and maybe that would impact your testing approach methodology. So just kind of to level set us a little bit about testing considerations and just kind of bring us back to the beginning. When we think about testing, what are we looking for? Right, we're looking for very low levels of contamination, generally a 1 CFU or colony forming unit in a 25 to 375 gram sample. That's obviously, there are other exceptions. Leafy greens, I know, just have, talked about a 1,500 gram sample. Some people in the dry fruit or spice range could see more something that's like a 750 gram. So we do see variations, but in general, you're looking for very, very low contamination in what can be a really, really big sample. Most current rapid methods still require a growth step. So we're still critically involved with, is something growing? So it goes from that one cell to actually something that's generally more around 10,000 cells. So we've got to get that one cell to grow up to about 10,000. And this is where it connects to what we were talking previously about, well, what impacts growth, right? Well, we just went through a lot of that. A lot of things can impact growth. Injury, competition, limits to biology. Maybe that organism just doesn't grow as fast. Stress, certainly, and there's an abundance of different things that could be stressors to these organisms. What types of nutrients there are available in the actual enrichment broth, and certainly time, right? So all of these things are testing considerations, not just for your product or for your sample, but also in the development of different test methods. To just kind of make this easy to understand, I could think about this, game that most people played when they were a kid, Hungry Hippos. The goal of the game is to get as many of the balls of food, right, that you can against your competitors. So faster players, so if you're just faster at hitting the little trigger so you eat more food, you'll win the game, right? You've got more faster. So if you have a faster organism that just grows faster than its competing microflora, you have a better shot at getting to that 10,000 cells, right? So you'll get the limit of detection faster. If one player is given more time, perhaps I say the blue hippo gets to start 30 seconds before everybody else, well, there's a really good chance that that one will also get more food, right? So that's another way that an organism could get up to limit of detection faster. Another could be the opposite, right? What if the blue hippo is actually broken or injured, right? So it doesn't work as well. And you're going to have a much harder time competing then with the other organisms that are trying, or the other hippos that are trying to get food. And that's the same thing that we see in enrichment dynamics. So it's just a quick and easy way to think about what's happening in my enrichment broth and why does time matter, right? Or why does nutrient availability or growth rate matter? Well, these are why. So if we want to think about visualizing an enrichment, so I have talked about media, and if you can kind of just imagine broth, it's a nutritive liquid generally, where we're growing things up. Other than we know we're putting our sample in that, we can't really see anything. So how do we visualize what's happening in an environment, and why does that matter for method development and detection? So if we think about this, is that all of our common detection methods, and these include the rapid ones, they're usually designed to identify one organism or one group of organisms. But enrichments are actually quite complex, and it's not just that organism that's living there. So the enrichment media is a very, very critical piece of any method that is still reliant on getting growth. So it will determine what grows, it inhibits what you don't want to grow, inhibiting them from getting the nutrients that your organism needs. It can help resuscitate the organisms and bring them back to rapid life. And you certainly can help minimize inhibitors in products and samples. And unfortunately for us, the food industry and food is quite complex compared to if you look at our clinical kind of compatriots where they are testing blood or urine. It's very simple compared to a chocolate cake or complex soil, fruit and vegetable sample. Enrichment protocols, these growth medias, are often then paired with detection technologies. And that's where you get into what we generally talk about with rapid methods. So PCR-based methods, immunoassays, LAMP, or biosensors. These newer, and very technologically advanced techniques are paired with still this growth phase. That's not always the case. There are newer detection methods that are coming out, which are very promising. But still today, almost all rapid methods are still reliant on some sort of growth phase where we're looking at media production and really how do we grow up organisms. And that is very, very much connected to the state of the organism and the ability for that organism to grow. So how do we see it how do we understand better what's happening in enrichment? Well, that's where we have the burgeoning growth in the area of genomic testing that really gives us a glimpse into the unknown, right? So genetic information on everything that's present, not just the organism that we're looking for. It can be based on DNA or an RNA approach. When we do some of these newer genomic tests, whole genome sequencing, we'll go through those in a little bit. We can target pieces of a genome, or we can completely assemble all the genome. We can eliminate culture bias. So we've talked a little bit about how hard it can be for some organisms to grow, but the realities are is that we're also limited in our ability of understanding what's there to actually know how to grow it. So with some of these newer technologies is that we eliminate that culture bias. Simply the idea of, well, we know what to grow. We know what's growing because we know how to grow it, but maybe there's something there that won't grow in the conditions that we've created. We can complete this before an enrichment, so effectively eliminating all of the culture bias and really potentially eliminating some of the stress considerations. Or we can do it after an enrichment to better understand how that changes or even throughout, right? How does that microbial population of our target, as well as competing organisms, change throughout the time? To do that, these tools are often what we would call whole genome sequencing options. There's shotgun sequencing, it's a whole nother webinar, so let's go over it real quickly. But long read sequencing, 16S or 18S and ITS, which are just certain gene targets that are between bacteria and then also with eukaryotes or fungi. Multi-locus sequence testing, so only targeting a certain set of genes that we can then compare. Sanger sequencing, which this is a slower version to get the kind of whole sequences. So there's just a lot of different ways that you could approach whole genome sequencing options. It's very rapidly evolving. It's really, really exciting. Right now, as of today, right, sensitivity is too low to be used as a detection method. So effectively, it's still not capable of picking up that one cell in your sample. So if the sample sizes are 375 gram or the like, is that we can't get down to 1 CFU level, which is why it hasn't displaced our normal detection technologies. Also, it's more expensive and more time consuming than our regular detection methods. But what we are seeing is very rapid evolution in this space where we know where the industry could be going, and it's really, really interesting. But right as of today, how else can we use whole genome sequencing to better understand our products, our investigations? You hear a lot about it with strain tracking, with different outbreaks and surveillance. Also, if you're doing spoilage studies, to do microbiomes to better understand what's going on in your environment. So we're seeing whole genome sequencing come into the industry in more regular testing quite often now, but it's not yet for our normal pathogen kind of detection due to sensitivity, cost, and time. But what it can do is help us see what's going on in an enrichment. So we talked, about thinking about an enrichment as this diverse ecosystem of lots of different organisms. And so this is actually an ice cream sample that has been analyzed using whole genome sequencing for the enrichment broth over time. It gives you a really cool snapshot of what's happening in this broth. So important, I know it's really difficult to see, but the important pieces here are the dark blue. You can see my cursor. The dark blue gets progressively higher until at like 32, 36, 40, 44 hours, 48 hours. It represents the predominant organism in the enrichment culture. But the important piece to look at is the diversity that you see earlier on in the enrichment. So if we had a method that let's say we tested this at 16 hours, we have a pretty low chance that we're going to find our target, which is down here at the bottom. So we have a whole bunch of other stuff. And you can see all these other organisms and the groups of them change over time as the enrichment continues. And so this just allows us to better understand what's happening, within this broth and what's happening in this method, so that we can better understand how to balance time as well as getting our optimal sensitivity. So on a produce-related study, what we can often find on the background of, in many different produce items, but with lettuce, is that you can often find 10 to the third to 10 to the seventh CFU per gram on a sample. So it's a lot of diversity of bacteria on these leaves. The detection methods that we run certainly can tell us an answer, but it doesn't give us the whole picture. It gives us the end result, right? So we know whether or not a certain thing is there. What it doesn't tell us is anything that's going on in the broth, and how that organism is responding and how fast it's growing and whatnot. Validations of our detection methods are built off of lab conditions. Obviously, we're doing this work in labs, and not necessarily real world conditions, which can be quite diverse, especially when we think about that within produce, is that produce has an amazing plethora of different types of conditions and environments that could really challenge a validation. But what we try to do in validations, and these would be inclusive of, ones performed in labs or with AOAC or something like that, when you're comparing different methods, is that they're designed to try to capture some microbial stress so that they're not in their optimum state. But we're obviously limited to practical lab protocols, otherwise we'd get nothing done. Validations don't specify what strains specifically to study, right? So we saw that Salmonella information from that study that showed a lot of variation between serovars as well as intra-serovar variability. Just like people, right? These organisms can be in different states or have different optimizations, and so you do see variability within an organism, certainly. But validations don't necessarily control that. So you could have one AOAC validated method against another AOAC validated method for the same thing, and they had a very different strain profile, how many and what they looked at to actually get the validation in place. So this study itself was designed to evaluate the impact of cold stress, or what was really a normal validation type stress for a method platform. So that was with using E. coli O26, which is one of the top 7 S-Tech organisms, and then obviously the background microflora that's normally found on lettuce. It was also an opportunity to allow us to see whole genome sequencing and really see what's going on in the enrichment. So not just do we get to the detection that we want with the cold stress or without it, but also what else is happening in the enrichment. I just mentioned it was used in an unstressed and cold stress condition and inoculated at high, medium, and low levels, which I'll show you right here. So overnight culture of S-Tech O26, so E. coli O26, and at 10 to the 8 CFU is then inoculated at different levels. A low 1 CFU per sample. These were 375 gram samples. Medium 10 CFU per sample, or high at 92 CFU per sample. And then we had 2 conditions, so unstressed and stressed conditions. So we did some on-product cold stress, which effectively means we just inoculated the organisms and kept it at 4 degrees Celsius for 48 hours prior to enrichment. So it also stressed the background microflora, and then also allowed the organism to attach to the lettuce itself. And then we also did an unstressed condition, which was just the straight bacteria on direct inoculation and then enrichment, so no time in between. So we just took that inoculum, inoculated the lettuce samples, used pre-warmed media, and went straight into the method. We use 2 different detection platforms. They're both PCR based, so BACS and BACGene, at different times. So you can see here, you have a 4-hour, a 6-hour, 8-hour, 10-hour, and a 16-hour. And you can start to see as each of the platforms and each of the conditions started to find it. So the important thing here really to recognize, I know it's a lot of data, is that at the time where the methods were validated, which is at 10 hours, everything was found, whether it be stressed or unstressed, but you can see there's diversity between the methods at different times. We also did, at each of those sampling points, microbiome analysis. So we took aliquots at 4, 8, and 16 hours in this case, and we immediately stopped everything from growing and created a frozen sample of that enrichment so that we could do this whole genome sequencing. Obviously, there's a lot of data on here and you can't really see, but the purple color is the target organism group. So not just the actual organism itself, but the group of E. coli. And then the dark black, color is actually a synthetic construct that was leftover reagents from the actual sequencing itself. And this was used using an Oxford nanopore technology, which is a long read sequence technology. Important thing really that I just want you to bring home from here is that the diversity of the organisms changes between the stressed and unstressed conditions, and it certainly changes over time within that condition. So you see the populations are quite diverse at the 4 hours on stress. You have a lot of different species that are represented. And then as time goes on, it actually starts to get less and less diverse, right? Until you have a much higher amount of your E. coli than you did when you were starting. The thing that was interesting with the stressed conditions is while we had no issues with detection of the actual target organism, it did very much change the diversity that we saw even initially with the background microflora, as well as each of these you can tell have a lot more synthetic construct. There's actually more of an artifact of not things got growing as well. So lower amount of DNA at the stressed conditions than the unstressed conditions. So the distinct population differences, the relative abundance effectively is what that graph showed you, and the composition was seen between the stressed and unstressed conditions. A lower amount of DNA in general for all organisms was recovered from the stressed condition versus the unstressed. All of the sequencing runs were using the same amount of reagents, so it did actually allow us to understand better if there was a difference between the overall copy of genomic DNA that was in the sample. Obviously, with validations, most people are doing cold stress, and it impacts the native microflora as well as the target organism. And that does change the microbial, the enrichment dynamic of what's there and how fast they grow. So it can either help your organism, like if your organism is not as susceptible to that cold stress as the background microflora, you could actually create a condition where it's easier to find your target in a validation study with cold stress, or vice versa, right? So you could see the opposite with your organism maybe is much more impacted by cold stress, and your microflora is not. Effectively at that point, your microflora can get way more food and outcompete your target organism. Diversity was certainly highest at the early stage of enrichment, and the 4 hours was the first one where we did the microbiome analysis, but we did quickly see that start to change. And that was consistent also with what we saw with the ice cream sample. If you recall, in the very beginning, stages and hours, is that you had many, many different colors, and then over time, you start to see only your target organism. The samples for lettuce with the Pseudomonas was one of the biggest organism groups. It significantly decreased from 4 to 8 hours, which is great to know because it's a very robust organism. Overall, E. coli populations, including our target, which was O26, grow to substantial proportions of the total population between 8 to 16 hours. So just in summary, to kind of connect very high level of a lot of topics that we covered in this time, is that when we look at microbial stress and injury, it can definitely have major implications for food samples and for testing, because our environments of which we produce food are not stagnant. They do change. And organisms can either be selected for or have to adapt to those environments, and that can very much impact testing results. They can obviously change day to day or sample to sample. They can change minute by minute depending on what your environment is. The key is when you're looking for methods and really evaluating different technologies, we want to balance the speed that we need with also the understanding of bacterial stress responses and potential injury. What we don't want to do is pay for a test and miss it because we maybe could have found it 2 hours later because our organism was stressed. So a lot of this all is a very complicated interplay of understanding our environment, our samples, the organisms, and certainly the detection methods. All in all, long story short, validations are complicated. They are not necessarily equitable to just look across the board and say they're all equal, because there's a lot of variability. Like I said, it really very much can depend on the strains that were included in the study and how they're conditioned and what stress they may have used for that validation. We also need to consider, our process or our product while optimizing enrichment conditions. It's just like a verification study that you might do to show that yes, I for sure can cover salmonella from this matrix that of a product. It's not going to impede its growth. But also think about, is there something specific in my processing environment that actually could be selecting for organisms that may not perform the way that would be expected in the validation? And it's not just the target organism you need to think about, right? If we think about the enrichment profiles that we saw using whole genome sequencing technologies, that there's quite an abundance of different organisms that might be there. And depending on what's there and how stable those are, they very much can change our ability to find what we're looking for. Maybe they outcompete, they steal more food, maybe they steal more space. Maybe they actually could be antimicrobial or inhibitory to that other organism that you're trying to study. These are all things that are incorporated within method development and are things to think about when you're evaluating your testing. Another thing, obviously we use some pretty cool tools to be able to see what was going on in enrichment broth, and these whole genome sequencing type tools and genomic technologies are developing not just so that we can do these types of studies, but they're getting better and better with sensitivity and getting to a world where maybe we aren't growing things up anymore. And there's certainly some of those today that are starting to hit the market, where we're looking for a certain amount of cells in our starting sample as opposed to trying to growing them up to begin with. But in this situation, whole genome sequencing was able to show us a difference in diversity and quantity, right? So we could see not just what was there, but how much of it, how did it change over time. With that, I will thank everybody for their time, and we have some sources, but I will open it up for questions, I believe, right, Amy?
Yep, that sounds great. And I'm actually just going to launch a poll right now. I just wanted to get some feedback from our audience. We'd like to ask you if you would like someone from Eurofins to follow up with you after the webinar. So go ahead and respond to that, either a yes or no, and we'll move into the short Q&A. So Joelle, the first question that came in here was, you referenced that rapid methods tend not to be able to detect low levels without a growth step. How sensitive are the rapid bioluminescence techniques?
Okay. In general, there's obviously a ton of different methods that are out there on the market, but in general, it's 10 to the 3 to 10 to the 4th. So 100 to thousands of cells that have to get to detectable limit. So 1,000 to 10,000 is generally what we say for people to think about. So unless you expect to have that high amount of your target organism in your sample to start with, we still are requiring a growth phase to get to that point. So that's, I think commonly lost in conversations about rapid methods is that we talk a lot about the very cool, fancy technologies that we use to detect. So whether that be polymerase chain reaction or PCR or immunoassays or MDS, is that, we talk a lot about those because quite frankly, they're amazing, right? The detection technologies that have evolved so that we can do this is quite remarkable. But we're still reliant on getting to that 10,000 cells, say. And that is very much still dependent on biology and the enrichment broths and the conditions of the organisms and getting there. So long answer, to a short question, but effectively what we generally say is 10 to the 3 to 10 to the 4th is a limit of detection for most rapid platforms that are out there today. There are some exceptions to that. Some newer technologies that are coming out may eliminate enrichments altogether. And I think where that is extremely promising in many ways, what we're still challenged on, especially in the food industry, is that the food matrices themselves are quite complex and our samples are quite large. So we want to get to that 1 CFU per 375 gram sample. Right now, it's very, very difficult to do that, even with these amazing technologies. So we're still very much reliant on the growth phase in most of our rapid technologies. Hope that answers the question. Not
okay. Perfect. Thank you so much, Joelle, for that. The next question that came in, are there any technologies that go straight to detection and avoid some of the potential delayed growth from stress?
Yeah, so I just kind of alluded to that there. We're starting to see a newer kind of phase of detection technologies that can do that. Again, the limitations at this point really fall more on sample prep and sample size. So we understand really well now the targets that we need to look for these organisms, and certainly PCR's detection. If you use PCR as an example, it can be one to a few copies of that gene that it can detect. So it's very, very, very sensitive. The trick is, how do you get a very messy, big sample into something that you can analyze to get to that level of sensitivity? So that's really where we still struggle. But there are some methods that are coming out with certain, certain matrices and situations, environmental samples, potentially, swabs, that have less complex, smaller samples that you can potentially do that. Go straight from the sample itself, straight to a detection platform, and still have the sensitivity that you're looking for. I think when we think about where is the industry evolving in the next 5 to 10 years, it will be that. The combination of better and better detection methods without these enrichment protocols, so it speeds up time, and then kind of paralleling that or complementing it with whole genome type sequencing technologies. We're just not there yet, but you can see where it's going.
Okay, perfect. Thank you. And it looks like we have a couple more questions here. What types of stress are usually used during validations of methods?
Yeah, so I mean, in general, there's a couple approaches that are usually used. The cold stress that was actually kind of described in the lettuce study is a common one, where it's a couple days at a refrigeration temperature. That's probably the most common. There's also some heat shock stresses that people can do. And I think what's really important is understand it's a great system to create, consistency amongst different platforms. So when you're trying to validate something in the lab, and it's great that we're not just going from an actively growing happy culture to do these studies. But what we also saw, right, when we saw this in the lettuce study, where it didn't really have much of an impact on the actual target organism. Whereas if we look at research that's been done, especially in the produce industry or even in certain processing environments, is that the stress from the real world may not be reflective of those stresses that are happening actually, in a validation study. So all the more appropriate to really think about your product and your process and try to reflect what could be happening so that you get the most value out of your testing.
Perfect, thank you. And it looks like we have time for one more question here that has come in. Have you ever seen products or method fail at the lowest enrichment times and work at a later time?
Yes. So I think that there's some papers on that as well as just, actually industry experience where, sometimes the difference between a positive and negative could be an hour or 2 hours. When you're running a sample that's less rapid, maybe you're doing an 18 to 24-hour enrichment protocol, it's probably not something you'd see because you'd get far beyond that limit of detection that we talked about, that 10 to the 3, 10 to the 4 CFU. But when you combine that with a stress condition and the most rapid enrichment protocol, you really are starting to butt up against the limits of biology. And it is theoretically possible, yes, where you could miss it, and we have seen that, where you could take a sample at 10 hours and not find it, and then find it at 12. So it's certainly possible, and I think even one of the sources in this slide deck on the source page has a study that was done, independent researcher that found that as well, looking at different platforms where certainly you could find it, but not necessarily the shortest time. So that is something to think about when you are running your method and choosing how you want it run with your lab.
All right, that sounds great. And it looks like we are going to wrap up our webinar and the Q&A session. Again, I want to remind everyone that a recording of the webinar, along with a copy of the slides, will be available within 3 business days. And thank you again, Joelle, for your presentation today. And thanks everyone for attending.
Thank you, everybody.
Thank you.